Okay, think about this for a second. Inside your body, right now, trillions of microscopic machines are constantly at work. They're building, repairing, and basically powering everything you do. We call them proteins, and in this explainer, we're doing a high-speed tour of their architectural blueprint. Think of it as a crash course study guide to how they're built, from a simple string to an incredibly complex machine.
So here's our game plan. We'll start with the overall blueprint. Then we're going to build a protein from the ground up through 4 distinct levels of architecture. We'll go from the primary chain to secondary helices, the tertiary 3D fold, and then the quaternary complex. And finally, we'll get into why getting this folding process exactly right is, quite literally, a matter of life and death.
Just look at these intricate structures. They're all proteins. And the big question we're tackling today is how? How does a simple, one-dimensional line of genetic code manage to create such complex, functional, three-dimensional machines like these?
Well, to figure that out, we have to start thinking like an architect. We're not just going to look at the finished building. Nope, we're going to follow the construction process step by step, from laying the foundation all the way to the final working structure.
And in the world of protein architecture, it all happens in 4 fundamental levels: primary, secondary, tertiary, and quaternary. Each level builds right on top of the last, adding a whole new layer of complexity. This is our roadmap. Let's get into it.
So, let's start at the very beginning. Level 1. This is the raw material, the fundamental sequence that's going to dictate everything that comes after.
So the primary structure, what is it? It's really just the linear sequence of amino acids. Think of it like beads on a string. Now, what's crucial here is that this specific sequence comes directly from your DNA. And for our study guide, here's the first key term. These amino acids are linked together by incredibly strong covalent peptide bonds. They form a tough chain called a polypeptide.
If you look at this diagram, let's just focus on that top part for a second, the primary structure. You can see it's really just a list of parts, all in a very specific order. All the potential for that final complex shape is hidden right here in this simple sequence. But right now, it can't really do anything at all.
All right, let's move on to the next level. This is where that simple chain starts to get some basic shape, some local framing.
Secondary structure is all about local folding. Patches of that long polypeptide backbone start interacting with each other. And the key force here is not those strong peptide bonds anymore. This time, it's all about the much weaker hydrogen bonds. And they're forming between atoms in the backbone itself, not the unique side chains. That's an important distinction.
And these hydrogen bonds create 2 really common, really stable patterns. You've got the alpha helix, which kind of looks like a coiled spring or a spiral staircase, and then you have the beta sheet, where the chain folds back and forth on itself to form this flattened, pleated sheet. You can think of these as the standard framing elements in the protein world.
Okay, now we're ready for the main event. This is where all those helices and sheets and all the little bits connecting them finally fold up into a single, functional, 3-dimensional object.
This right here is the tertiary structure. It's the final overall 3D shape of that one polypeptide chain. And the key players have changed again. We're not talking about the backbone anymore. Now it's all about the R groups, the unique side chains on each amino acid. They're the ones driving this final, complicated folding process. And these R groups interact in all sorts of interesting ways. You have hydrophobic groups, the ones that hate water, that bury themselves deep inside the protein's core. Then you can get these super strong disulfide bridges that lock parts of the chain together like a staple. And then there are weaker forces like ionic bonds and these subtle van der Waals attractions that help fine-tune that final shape. They all work together to hold the masterpiece in place.
And here's the result. Looking at the tertiary structure in this diagram, you see it. The single polypeptide chain has now folded into its final functional shape. It's not just a string or a collection of parts anymore. It's a specific tool, perfectly designed for a specific job. For a lot of proteins, this is the end of the line. This is their final form. But for some proteins, there is one more level of organization. Because sometimes one functional unit just isn't enough. Sometimes you need a team.
That's where quaternary structure comes in. This is when multiple already folded polypeptide chains, we call them subunits, come together to form one single much larger protein complex. You can think of it like assembling several finished components to build a much more sophisticated machine.
And the perfect example, the one everybody learns, is hemoglobin.
You know, the protein that carries oxygen in your blood. It's actually made of 4 separate subunits, 2 alpha chains and 2 beta chains, and they all have to work together perfectly. It's this complex, multi-part structure that lets it so efficiently grab oxygen in your lungs and then drop it off all throughout your body.
So, we've gone from a simple chain to a complex, multi-part machine. But why is it so incredibly important that this whole architectural process goes exactly right every single time?
Because the stakes are incredibly high. A protein that doesn't fold correctly isn't just useless, it can be actively dangerous, even toxic to the cell.
You see, when a protein misfolds, it obviously can't do its job. But what's even worse is that these misshapen proteins can get, well, sticky. They start clumping together into these toxic clumps called aggregates. And these clumps are directly tied to devastating neurodegenerative diseases like Alzheimer's and Parkinson's.
This right here is the single most important takeaway from this whole thing. In biology, structure dictates function. It's a protein's very specific 3-dimensional shape that allows it to do its job. You change the shape, you destroy the function. It's that simple. So we've seen how a perfect genetic sequence folds into a perfect structure that builds life as we know it. But that leaves us with a pretty critical question to think about. What happens when the error isn't in the folding process, but in the original blueprint itself?